Okay, I'll try hold it close. Does that help? More importantly, Vatsan, if you talk, does it pick it up?
Yeah, I think it works.
I'll take it a few minutes.
I wasn't sure what the rules are. Maybe I could take it off.
Can you go ahead and say something so we make sure it's...
Can folks hear me okay?
Yeah,
Okay.
Your microphone seems to be on. This one, less so. So as I said, I'll go ahead and start introducing Vatsan. I'll try to talk loud so hopefully you'll hear me. So I first got to know or became aware of Vatsan in 2014, 2015 when I was running a campus-wide biodesign and synthetic biology search. And in that search, Vatsan really emerged in that first year. We ran the search for 2 years, and actually altogether it led to recruiting 3 of our really talented young junior investigators. But in the first year of that search, Vatsan really emerged as the top candidate, and I'll come back to why that is here in a minute. But let me introduce you to Vatsan first. He got his undergraduate degree actually in chemical engineering, so Vatsan has a chemical engineering background originally, at the University of Baroda in the Indian state of Gujarat in 2000. And he actually worked on process design for petroleum refineries at that point. Spent a little time as a sales executive, as I understand it, before he then went to a master's degree program at the University of Science and Technology in Rolla, Missouri. So I've actually been to Rolla before, having spent 10 years at Washington University, and all I can say, Vatsan, is that must have been quite a culture shock to have transitioned from India to Rolla. And he completed a master's degree there in 2003, went on to a PhD program at University of Washington, Seattle in biochemistry, and it's there that he really developed interest in what he's working on now, doing his PhD with David Baker, and working on purely computational methods, really at that point working on the beginning of what has now become Rosetta Fold, or some of you know about this in the context of AlphaFold 2, so the 2 programs that have really revolutionized the ability to predict protein structure. And Vatsan worked on early stages of that, and actually in 2008 led the team from David Baker's lab that won the CASP competition that year. So Vatsan's PhD there also focused on computational methods where they figured out how to phase X-ray crystallographic data by combining low-res NMR data with homology models and computational models. And after that purely computational and really successful PhD, Vatsan made a big switch and then went into experimental work by doing a postdoc with George Church at Harvard. And so it was really there at Harvard that Vatsan developed an interest in allosteric transcriptional regulators that he's continued on in his work now at the University of Wisconsin. So the hallmark really of Vatsan's research, I would say, and really what led him to stand out so much in that search the first year we conducted it, has been that he uses biodesign, mutational approaches, and computational approaches, not just as ways to build new cool things by synthetic biology, but really to understand fundamental principles. And so to understand principles of allostery, and to use these tools not just to make things, but also to generate knowledge. And I think that's what's made Vatsan, I think, really an enjoyable colleague to have around, a great person to talk with. He's always sort of pushing the cutting edge of what can we do to push the new frontier in science. And I think that's what I've really appreciated about having him as a colleague. So today, Vatsan's actually going to tell us about one aspect of his work, a little different than transcriptional regulators, that concerns bacteriophage and bacteria-host recognition specificity and how they've used deep mutational scanning to do that. So as you listen to Vatsan's talk today about bacteriophages, I'll leave you with just one number to contemplate as you think about the significance of this work. And this is a number I just learned recently and found unbelievable. I think you'll find it unbelievable. So we all know that the speed of light is like 186,000 miles a second, and that a light year is something like 5 trillion miles. So that seems like an incomprehensible number. But what's truly incomprehensible is that if you took all of the viruses on Earth, and those are almost all bacteriophage, and you line them up one after another in order, they would reach 100 million light years. So contemplate that as you think about the significance of Vatsan's work today. And with that, Vatsan, I'll turn it over to you.
Well, thank you, Bob. Can people hear me okay? Super. Before I get started, I want to acknowledge people in my lab. It's such an honor and a privilege to be their mentor. They're all very talented people, and they could have really joined any lab they wanted, and they joined mine. And so I'm very fortunate for that. And to discuss science with them and to brainstorm ideas with them has been such a big privilege for me. Okay. So as biochemists, our goal is to understand the molecular rules of life. And to achieve this goal, we pose two types of questions. The first type of question is one of mechanism. And the mechanism question seeks to explain how different biomolecules interact with each other to carry out some cellular process. The second type of question that we pose is one of function. And the function question seeks to explain what is the molecular basis of function. In other words, if you look at a protein, what is the role of every residue of a protein towards its function? Now, I would argue that over time, we've become really good at understanding the mechanism of different cellular processes. In other words, we've become good at wielding a scalpel, so to speak, to sort of deftly peel off layers and understand biological mechanism. So pick any cellular process, transcription, translation, signaling, transport. We understand these at great depths because the reductionist approach to understanding biological mechanism works really well. Now, we've also made pretty major advances in understanding the molecular basis of function, right? And so structural biology tools such as cryo-EM, NMR, crystallography have played a pretty major part. And yet, I would argue there's a lot that remains unknown. And the reason for this is structural biology tools give you, a structure, right? And so going from structure to molecular basis of function, that's not really a straightforward process. But let's pause here for a second and ask, well, what do I mean by molecular basis of function, right? So the overall function of a protein is simply the collective output of the function of every residue of a protein, right? And so when I say molecular basis of function, what I simply mean is what is the role of every residue of a protein towards its specificity, towards its activity, towards its affinity to other biomolecules, stability, allostery, etc. Now, if we want to understand what is the molecular basis of protein function, we have to understand how sequence variations impact protein function. In other words, how systematic changes to protein sequence impact protein function. And so this question, how do sequence variations impact protein function, this kind of gets to the root of a problem in protein biochemistry that we often tend to ignore. And that is the bigness, quote unquote, of protein sequence space. And so what do I mean by that? What decades of biochemistry and genetics has taught us is that a single mutation to a protein or two mutations to a protein can have profound impact on function. And so let's examine the sequence space of these changes, right? So let's imagine that you have a 100 amino acid protein, right? And what you wanted to do is you wanted to systematically mutate every residue of the protein to all remaining 19 non-wild type positions, right? So protein-wide single site saturation mutagenesis. And so what's the sequence space of these variations, right? So it's 100 times 19, approximately 1900 mutants, right? So this is a fairly large number for traditional biochemical assays. Now you can take it a step further and say, well, what does double-site saturation mutagenesis look like, right? So pair of residues and all 19 amino acids at a pair of residues, right, across the entire protein. And so the sequence space of these changes is upwards of 1 million, which is a staggeringly large number for biochemical assays, right? And so you can do a triple-site saturation mutagenesis, and so it's 1 billion sequence combinations, right? And so the sequence space explodes very quickly. Now, nature, of course, it samples all the mutations at all positions all the time, right? And so you already seen evidence of this when you blast search your favorite protein. So when you blast search your favorite protein, you might find, hundreds to thousands of sequence matches, which are anywhere from 90% to 20% sequence identical to each other. Which then begs the question, well, how are these sequences functionally different from each other, right? Do these sequence variations encode new activities, new specificities, different stabilities, different allosterities? So we simply have no answers to these questions. And so I would argue that if we want to answer the question, what's the molecular basis of protein function, we have to start making functional measurements at the scale at which nature samples them. Or we have to start making functional measurements at the scale at which we're discovering new protein sequences from genomic databases, right? And so this idea of picking one wild type or one other mutant is just simply not going to cut it. And so I want to sort of illustrate this idea with a simple example here. So let's imagine that this is some sequence function landscape, right? So on the x-axis, you have some sequence variations. On the y-axis, you have some measure of function. So when you pick out one or a few sequences to biochemically characterize, what you really see is a small sliver of the sequence function landscape. And so the example that I like to give is this old parable of 5 blind men and the elephant, right? And so there are 5 blind men who touch different parts of the elephant, and they want to figure out what the elephant looks like. Now, if they touch these local regions, so they have really no idea what the elephant looks like, right? And so when you biochemically characterize one or a few sequences, what you see is the small sliver of the sequence function landscape. Now let's say instead of 5 blind men, you had 50 blind men, right? And so these 50 blind men, collectively have slightly a better sense of what the elephant looks like. And likewise, as we start sampling more, sort of functionally sampling more, you start seeing the peaks and the valleys of the sequence function landscape. And let's say instead of 50 blind men, you had 500 blind men, right? And so now they have a pretty complete sense of what the elephant looks like collectively. And likewise, as you start functionally sampling more and more sequences, you have a pretty full sense of what the sequence function landscape looks like. And then you can learn the molecular rules of how the protein functions based on how these sequence variations impact function. So which then brings me to what my lab does. So the overall goal of my lab is to generate and understand these sequence function landscapes of proteins, and to then use that information to figure out the fundamental molecular rules that govern protein function. And so to do this, we introduce mutations in very high throughput, and we make functional measurements in very high throughput. So the question is, well, how do we make functional measurements in high throughput, right? And so the answer is to use next generation sequencing, also known as deep sequencing. So I'm going to do a small detour for a couple of slides, and then we'll get back on track. So in the field of computer processors, there's something called as Moore's law. And so this was named after Gordon Moore, who is the former CEO of Intel. And what Moore's law states is that the power of computing doubles roughly every 2 years, or the cost of computing halves roughly every 2 years, right? So if you think about that for a second, that is explosive technological growth, right? And so besides computer processors, there's really no other technology that matches the growth of Moore's law, except next generation DNA sequencing. So next generation DNA sequencing not only beats Moore's law, it beats Moore's law by a mile. And so here is 2005 when next generation DNA sequencing was sort of roughly invented. And so which resulted in a precipitous drop in the cost of sequencing. And that ushered in what I like to think of as a sequencing-driven revolution in biology. Now, what does this have to do with protein sequence function landscapes? I'm going to get to that in a minute now. Now, the sequencing-driven revolution in biology really unfolded in 3 stages. The first is the first stage is what I like to think of as compositional measurements, right? So here we use sequencing to measure the, sort of sequence genomes of different animals to figure out molecular phylogenies. We sequence human populations to understand the genetic basis of disease and other traits. We sequenced microbiomes to figure out the composition of bugs that live within us. So this is the compositional phase. So in the second phase, this is what I like to call as the observational phase. And so here we started using next-generation DNA sequencing to measure endogenous biomolecular structure and function, right? So there's an entire alphabet soup of different seqs, such as RNA-seq, ChIP-seq, methyl-seq, ribo-seq, which measure different cellular processes, right? Chromatin state, transcription factor binding sites, genome structure, etc. The third phase is what I like to call as the perturbational phase. And here we started using next-generation DNA sequencing to introduce mutational perturbations and to make functional measurements with next-gen sequencing. And so this is where I'd say my lab makes a contribution. And so it was roughly around 2015 when just a number of sort of gene editing technologies and other approaches started coming out. And what we figured is if we can combine the ability to introduce mutations in high throughput to make functional measurements in high throughput, well, in principle, we can reconstruct these sequence functional landscapes. And so that's what we did. Now, DNA sequencing turns out to be an ideal readout for these kinds of measurements because the volume of DNA sequencing scales with the bigness of protein sequence space, right? So a typical sequencing run would produce hundreds of millions to a couple of billion reads, right? And so if we can somehow couple biomolecular function to sequencing, then we can exhaustively map the mutation function landscape, right? And so the question is, how do we make this connection between biomolecular function and sequencing? And so to do this, we take a few different approaches in my lab. First is we use a reporter system. So different gene variants or protein mutants activate the reporter to different degrees. And so we can sort the reporters into bins based on fluorescence and then sequence the bins. Or we can use a barcoded RNA. So different transcripts are barcoded, and then we can do cDNA synthesis. So these are all sort of reporter-based measurements. So the second class of measurements is where we can link the protein variant to cell fitness. So some variants enhance fitness, some variants hurt fitness, and so you can quantify the effect. The third approach, which we're beginning to use, is to use single-cell RNA-seq, right? And so here the goal is to ask how every variant impacts the transcriptomic state of the cell in its entirety. And so we use these general principles to study 3 questions in my lab. So the first question, as Bob mentioned, is protein allostery. And so this is, allostetic proteins are proteins where if you introduce a perturbation at one end, you see action at a distal end of the protein. And so what we showed last year was that allosteric hotspots, which are residues critical for allosteric signaling in proteins, are not conserved necessarily. And what we also showed is that what is commonly thought to be the shortest link between the allosteric site and the active site, which is thought to contain the allosteric hotspots, that turns out not to be the case, and the allosteric hotspots are more distributed, at least in a couple of examples that we looked at. The second problem that we were working on is we're asking, what is the functional impact of variants in disease-relevant genes? And so the goal here is to ask if we can use high content measurements like single-cell RNA-seq to measure the functional impact of disease of variants of mutations in disease-relevant genes. Now, what I want to do today, though, is focus my talk on the third part of my lab's work, which is understanding the molecular code of bacteriophage-host interactions. Now, as Bob mentioned, bacteriophages are pretty remarkable creatures. And so just to put a number on that, the estimates are there are 10 to the 31 bacteriophages on Earth, so which is kind of a mind-boggling number. And you can imagine that the arms race between bacteria and bacteriophages are sort of the longest evolutionary experiments conducted by nature, so to speak, right? Now, bacteriophages have high sequence diversity and genome mosaicity. And sort of this enables them to adapt to different environmental niches. It also creates this tight link between sequence variations, and phage fitness. Now, in fairly simple terms, the life cycle of a bacteriophage, in this case a lytic bacteriophage, really involves 4 simple steps, right? The first step is the phage absorbs on the host. The second step is the phage delivers its genome into the host. The third step is it hacks the host machinery and makes many copies of itself, sort of replicates inside the host. And the fourth step is it lyses the host and then goes on and infects other hosts, right? And what really got us sort of interested in this in bacteriophages is the societal grand challenge that we face with antibiotic resistance, right? And in just a couple of charts that I'll try and illustrate the scale of the problem. And so this was a study that was done by the UK government a few years back, which showed that if multi-drug resistant bacterial infections, the problem is not addressed, the number of deaths from bacterial infections is likely to surpass cancer in the next 2 decades or so. And what was particularly alarming are these 2 charts. And so here is a chart that shows when different drug molecules were introduced in the clinic over time, over the past 5-7 decades, and when the first resistant bug emerged in the clinic. And you'll see that in the 50s and 60s and 70s, it was typically about a decade when a drug sort of lasted in the clinic. But more recently, as soon as you introduce a new molecule, there is spontaneous resistance to the antibiotic. Now, the second chart here shows that the number of antibiotic molecules that are coming through the pipeline, right? And so this is a dwindling number, right? And so we have this sort of double whammy where on the one hand, we're not discovering new molecules, and on the other hand, even if we do, they don't last too long in the clinic. And so we said, well, how about we come up with a completely brand new idea to address this question, which is to use phages for therapy. Now, of course, I'm kidding there because people have known or used phages for bacterial treatment for almost 100 years, right? And so here is an ad from the 1930s, which is this place called Le Laboratoire du Bacteriophage, where they were sort of providing phage treatments for intestinal diarrhea, E. coli infection, staph infection, etc. And so this place, Le Laboratoire du Bacteriophage, eventually became L'Oreal. And so this is the history of the largest cosmetic manufacturer in the world. Now, in the West, we discovered penicillin, right? And so we never looked back. Penicillin turned out to be a miracle drug, and we never looked back until now. Now, in many ways, I sort of think of the phage as the ultimate smart drug for a few different reasons. One, it's highly precise, which means that it can specifically target a microbe within a complex community while leaving the rest of the community structure intact, right? And antibiotics, on the other hand, kind of wipe the whole place clean. Now, phages are evolvable, which means that bacteria will inevitably become resistant to a phage as they do become resistant to small molecule antibiotics. But with the phages, in principle, you can take them back into the lab and re-evolve the phage to remain efficacious. But with an antibiotic, it's basically a dead end. It's auto-dosing in the sense that it only amplifies at the site of infection. And fourth is abundance, which is because there's practically a limitless supply of phages for any bacterium, right, or any known pathogen, right? So one can go back into nature and then use these phages as natural phages as therapeutics. Now, over the last few years, there have been a number of examples where phages have been used in the United States for therapy on compassionate grounds. And so the one that caught people's imagination was this case where Dr. Tom Patterson and his wife, Dr. Stephanie Strathedy, they were vacationing in Egypt. This was 3-4 years back. And he ended up getting a multi-drug resistant Acinetobacter baumannii infection, and so he could not be treated with the normal course of antibiotics. And so then he had to be flown back to San Diego, where they're both professors at UCSD. And so he was then given a cocktail of phages that was put together by Texas A&M, Naval Research Labs, and this company called Intralytics. And so he made a quick recovery, but then within a week, there was a resistant strain that emerged, and then the phages were no longer effective. And then they took the resistant strain back into the lab, and then they re-evolved the phage, or discovered new phages that were effective, and then it was reapplied. And he's lived, he's living a hale and healthy life since then. And there have been other instances where phages have been applied in the last 3 or 4 years for therapy. Which then begs the question, well, if phage therapy is really as awesome, well, why hasn't it been widely adopted yet, right? So there are a few fundamental challenges with the use of natural phages for therapy. First is they have a narrow host range. Now, I just mentioned this as an advantage a few slides back, but it also turns out to be a serious disadvantage because if you have two different strain variants of a same pathogen, a phage that might infect one may not infect another. Right. Second is the efficacy of traditional antibiotics is much higher than phages. Right. And there is a reason for this because phages exist in an evolutionary equilibrium with their hosts. Right. And so a phage that is really good at killing its host will itself perish. Right? So natural selection kind of selects for these mediocre phages. And so if we want to achieve high pathogen clearance, well, we have to start removing these evolutionary restrictions. Third is the inconsistency of phage cocktails. So phages are normally delivered as a cocktail of unrelated phages, and sometimes their life cycles kind of clash with each other. And so this results in inconsistency in the performance. And fourth is bacterial resistance. And like I said before, bacteria do become resistant to phages, and we don't have the methods in place to rapidly evolve bacterium to phages to remain efficacious against bacteria. And so the goal that we set out for ourselves is to ask, can we understand how natural phages work? And can we then use that information to engineer synthetic phages against pathogenic bacteria? So what I'll do today is I'll sort of have 4 short vignettes that I want to talk about. First, I'll tell you a method that we developed called Oracle for high-throughput targeted genome engineering of phages. Second, I'll tell you how we used Oracle to carry out deep mutational scanning of the receptor binding domain. And third, I'll tell you early results on how we're using this tool to measure the function of metagenomic sequences that are attached, that are integrated into phages. And fourth, I'll tell you our early steps at making therapeutic phages. And so we decided to focus on the receptor binding domain. And so the receptor binding domain makes primary contact with the host receptor. And so essentially, we wanted to understand the sequence function landscape of the receptor binding domain because it governs both the activity of the phage in the sense how well it can absorb onto a host and the specificity of a phage, which is what are the different hosts it can stick to. Right. And so we chose this bacteriophage called T7. Now, this is a really well-studied bacteriophage. And so this is the entire receptor binding protein of bacteriophage T7. It's a trimer. And we focused on this little tip domain, which is at the extremity of the receptor binding domain. And so you might recognize this as an IG-like domain that nature uses over and over again to stick to different things. And so what we wanted to do was to do deep mutational scanning of the receptor binding domain. And so the idea here is for every residue of the receptor binding domain, we wanted to mutate it to all remaining 19 amino acids, right? So the receptor binding domain is 83 residues, so 83 times 19, which is 1577 unique sequence variants, right? Now, it turns out that there are methods to do random mutagenesis of phages using chemical mutagens or radiation, but there weren't methods to do targeted mutagenesis of particularly lytic phages. And the problem here is that you have to introduce these mutations in the phages as they're actively lysing their host, right? And so that makes it technically challenging. And so for instance, in the case of T7, it's about 20 minutes. So you have to sort of introduce these mutations onto the phage genome in 20 minutes. And so what Phil Huss, who's a very talented graduate student in my lab from the microbiology PhD program, he developed this method called Oracle, which stands for Optimized Recombination, Accumulation, and Library Expression. And what it does is it's a high-throughput, locus-specific phage genome engineering method to make large, unbiased library of phage variants at any target gene. And so what I'll do is I'll just sort of briefly explain to you how Oracle works in a few slides. So what we do is we remove our gene of interest, in this case the receptor binding protein, and then we introduce recombinase sites around that. So we make what's called as an acceptor phage. And we introduce a random sequence in between, and we have recombinase sites. And so this phage can no longer propagate, right? So here is simply data that shows that once you remove the receptor binding domain, the phage has an 8 log drop in infectivity, right? So this is lower being poorer. And once you reintroduce the receptor binding domain, so the phage is as effective as wild type, right? And so now we have this phages, which is these modified phages, which cannot infect. And so to make them infect, we provide the receptor binding protein in trans, right? So this is sort of complementation of the receptor binding protein. The second step is to now introduce the gene variants. And so the way we do this is we make this phage infect a population of cells, each containing a different gene variant. And so these gene variants are synthesized by using chip-based oligonucleotides. And so now what we can do is we can activate the recombinase inside each of these cells, and it sort of flips the variant into that cassette location that we introduced at the very beginning, right? And so we typically get about 1 in 1,000 phages recombined at this stage. Now, what is really critical with this step is to make sure that the receptor binding domain is provided in trans the whole time. And the reason we want to do that is we want to create an unbiased library of mutations that we can apply on different hosts for selection. And so by providing the wild type receptor binding protein in trans, we avoid any premature biases that might emerge in the library synthesis process. Now, the third step is to now enrich the recombined phages and to deplete the phages that have not recombined, right? And so the way we do that is we introduce Cas9 into the cells, and so the Cas9s now target that random piece of DNA that we introduced at the very beginning. And so the unrecombined phages are cut up, and the recombined phages are enriched. And so this is sort of an enrichment of the recombined phages as a function of time. And the last step is then we no longer provide the wild type receptor binding protein in trans. We make the phage express that gene variant, and so we have these population of mutated phages. And so here is just data that shows that here is the input plasmid library going in, and here is the output genomic library integrated onto the host genome. And there's a little bit of a skew coming out, but it's more or less unbiased. And so the advantage of Oracle is that A, it is locus specific, so you can pick any phage gene to introduce these mutations at. B, it's sequence programmable, which means that you're not introducing random mutations. You can pre-specify what changes have to happen, right? Third is that the functional throughput of this approach is 3 to 4 logs higher than a traditional plaque assay or liquid culture assay, right? And fourth, and this is an important point, is that it works on lytic phages. And the reason this is an important point is because the FDA mandates that only lytic phages, not lysogenic phages, can be used for therapy, right? So now I'll sort of tell you briefly how we use this approach to carry out deep mutational scanning of the receptor binding domain. So like I said before, what we wanted to do was we wanted to introduce all 19 variants at every residue, right? And so I'm going to sort of quickly walk you through how this works. So we want to systematically mutate every residue to the remaining 19 residues. So in this case, the first amino acid is a glutamine, and we want to change the glutamine to all 19 amino acids. And so you get 19 sequence variants at that position, right? And so now we walk over to the next residue. This is a valine. We want to change this valine to all remaining 19 amino acids. So that's another 19 sequence variants. And so you can walk through the entire length of the receptor binding protein, and you get to residue number 83. And so you sum it up, and so that's how we end up getting 1,577 mutants plus 1 wild type allele that's present in the mix, right? Okay. And so now we can generate this library using Oracle, and then this is where the next-gen sequencing technology becomes really useful. So what we can do is we can take this population of variants, apply them on a host, and simply use next-generation sequencing to count the number of phages that went in of a particular variant and count the number of phages that come out, right? So if a variant is deleterious, it'll be depleted. If a variant has no impact, its relative abundance will not change. And if a variant is beneficial to the phage, then you'll see enrichment. Okay. So I'll just sort of mention a couple of points here. One is the theoretical library size was 1577. The actual number of mutants that we found in our library generation process was 1573. So we had pretty good coverage of the entire mutational space. And so now we apply this library on 2 sets of hosts. The first set of hosts are susceptible hosts. So these are just 3 different E. coli strain variants. the other host is a host that lacks the receptor, right? So this is a resistant host. So what we'll do is we'll first look at the results from E. coli 10G. I'm going to sort of, before I zoom out, I'm just going to quickly walk you through sort of a couple of residues, and then we'll zoom out. And so here is the result of mutating the glutamine to all remaining 19 amino acids, right? And so what you'll see is that each block is colored based on a functional score, right? So, and here is sort of the range of the functional score. And so a white block is a mutation that drops out. So it's a deleterious mutation at that position for the phage. The reds are those that do survive selection. Now the wild type amino acid at that position, which is this, is normalized to a score of 1. And you can see that, this little block there, sorry, the gray circle there, that sort of marks the wild type position. And so what you'll see at this position is that it is tolerant to most substitutions, right? So aspartate, glycine, phenylalanine, and tryptophans are not viable at that position, right? And what you'll also notice is that there are a couple of positions that actually outperform wild-type glutamine at that position, right? So this is, now we'll just sort of walk over to one more position. Now this is the second amino acid, valine. Now this tells you a different story, right? And what you'll see here is that most positions are white, which means that they drop out of selection. So there's the alanine and the valine. Well, valine is the wild type amino acid. There's alanine and isoleucine that are viable, and cysteine to a lesser extent, right? And so now I'm going to sort of zoom out and look at the full map, but I just want to point out a couple of things about this map before we look at it. So these these alphabets here are sort of the topology of the receptor binding domain, sort of different beta strands, and the loops that link the beta strands. So B, C, etc. Right? And so now when we look at the entire sort of sequence function landscape, now I'll point out a few interesting features here. The first thing you might I want to sort of draw your attention to is this little part here. So the C-terminal end of the receptor binding domain is permissive to all substitutions, and this kind of made sense to us because it's a floppy little bit which has no role in either structure or function, and so it was not surprising that it could take a lot of mutations there. Now, what you might also notice is that the overall sort of fitness landscape is relatively sparse with peaks, which means that most mutations are deleterious to the phage. Now, what you might notice is that what was really neat for us was that we found a bunch of phage variants whose fitness was about 20 times or even more higher than wild type, which had a fitness which is 20 times higher than wild type. And what we can do is we can sort of zoom in and ask, well, what are these positions that are getting enriched, which have much higher fitness than wild type? And it turns out that these mutations are, arginines and histidines and lysides, which are all positively charged amino acids. And so one hypothesis is that these positively charged amino acids enable the phage to interact better with the negatively charged sort of phospholipid group of the outer membrane of E. coli. Now, what was also interesting was that there were specific regions of the receptor binding domain where these function-enhancing substitutions fell, right? Now, I'll point out later that this is sort of the exterior loop, but this is valuable information from the standpoint of phage engineering because this tells us where to focus our mutational efforts on to engineer synthetic phages for higher activity or different specificities. Now, so this is sort of the overall picture. What we can also do is sort of zoom in and look at specific sort of interesting local features, and I'll point out one here. So if you look at this little bit, we see this alternating patterns of positions that are tolerant and intolerant to substitutions, right? And then we said, well, what's going on here, right? And so we can go back, we can look at the structure again and see if we can potentially make sense of this. So this is the entire receptor binding protein. We're going to zoom into the receptor binding domain. And so the positions that I showed you in the earlier slide, the red positions are the positions where it was tolerant to many substitutions. The gray positions are positions where it was intolerant to substitutions. And what we could see here was that the C alpha of the positions that are intolerant to substitutions are pointing inward toward the core of the protein. And the red positions, the C alphas, are pointing out. So which means that any change to the gray positions essentially hurts the stability of the receptor binding domain. That's one likely possibility. Now, we then took the exact same phage library, and then we applied that on a different host, and this is BW25113. And again, this is sort of two different mutational maps sort of juxtaposed next to each other. And I want to point out that the patterns are different, which means that the phage is making different adaptations based on different hosts. You'll see that there are way more substitutions that are permissible in BW25113 relative to 10G, which also tells you that the selection bottleneck on 10G is narrower compared to BW25113. Again, we can zoom into one particular region of this map and see if there is an interesting sort of sub-story there. And so I'll point out this region here. Again, you'll see that there are fewer substitutions that are permitted on 10G, but many more substitutions that are permitted on BW25113. And to ask what might be going on here, we can zoom out and look at the structure again. And so that little part that I'd highlighted in the previous slide, it is this edge beta strand that kind of holds the receptor binding domain together, right? And so one hypothesis is that if in the case of 10G, any mutation, there is a certain orientation of the receptor binding domain which is sort of held together that might be required for the phage to infect 10G, but there is much more intra-domain flexibility that may be permitted on BW25113. In other words, it's likely that this is sort of a tight trimer, but this is maybe more of a splayed trimer, if you will. Now, again, I'll show you the data on BL21. This is, again, just a lot to take in, but just to point out that there are different patterns. Now, what we did was we just used the information from multiple selection experiments, and we asked, can we assign a role to every residue of the receptor binding domain? And so the roles were, is that position intolerant to substitution? Is that position tolerant to substitution? Or is it more of a specificity guiding kind of a position, right? And so this is based on the aggregate information from multiple selection experiments. And so again, as I mentioned before, the floppy C-terminal end, you'll see that it's tolerant to most substitutions on every strain. What you might also notice is that the hydrophobic core of the protein is, sorry, this arrow should be pointing here. The hydrophobic core of the protein is fairly intolerant to substitution, which makes sense from a structural biology standpoint. But what's neat is that these function-enhancing substitutions are at the very tip of the receptor binding domain. So these are, you can imagine, fingers that kind of engage the host receptor. And as I mentioned before, from the standpoint of phage engineering, this is valuable information because that sort of cuts down the sequence space for us to survey. What was also interesting was that these function-enhancing substitutions were found along the sides too, which leads one to hypothesize that maybe there are alternate poses of the receptor binding domain that could still lead to productive infection. Now, I'll sort of briefly talk about the results that we had from applying this library on a resistant host. And so what we were looking for was, a phage requires its host to infect. And once you remove the host, the phage, depending on what the phage is, it could have a significant loss in the ability to infect. And so what we wanted to ask was, can we identify gain-of-function variants? And so when I say gain-of-function variants is, once we remove the host receptor and when we apply the phage library, are there phage variants that can still infect the host that lack the native receptor? And so in the case of T7, it binds to lipopolysaccharide. And so we made a deletion mutant, which is called delta RFAD, which completely removes the LPS in E. coli. And here is sort of data that shows that with wild type T7, this is sort of a funny plot. This is an efficiency of plating measurement. And so if you're closer to 0, that means you have very high infectivity. And so here is almost a 6 log loss in activity of T7 wild type on a host that lacks that receptor. And so now we take the same phage library and we apply that on the host. And so what was really neat was we managed to identify a bunch of variants that could infect the host that was lacking the receptor. And again, you'll see these red points. Again, just to point out, these gray are data points which we didn't have in the sequencing experiment. And again, what you might notice is that the first thing you might also notice is that the the mutational map was really sparse this time, which kind of made sense because the wild type T7 has a hard time infecting this. What you might also notice is that these gain of function variants are again concentrated in those exact same regions that we had identified before. And one can sort of summarize the findings of this experiment with this alluvial plot. And so what this plot shows you is this is the wild type position, this is the substituted amino acid, and here is the topological location where those mutations were found. And so what you'll see is, and so the thickness of the line is a measure of the functional score of that variant. And so what you'll see is that most of the changes in the gain of function experiment were glycines that were mutated to histidines, arginines, and lysines, and most of them fell in this exterior loop FG, which is out here. And now we clonally validated these, so we take these variants and then we go back and apply them on the hosts, and what you'll see is all the three variants are able to infect the E. coli that lacks the host receptor. So in fact, this one actually outperforms wild type on that. So this one actually outperforms the others. And and that doesn't come at the loss of the ability to infect wild type E. coli, which means that they can infect both the deletion mutant and the wild type host. And so what did we learn from this? This is sort of a couple of quick lessons that I want to mention. That first, even a humble Ig-like domain has a pretty big story to tell, right? And every residue of that protein has a story to tell. And the phage is kind of remarkable in that it's able to make these host-specific adaptations, which again sort of speaks to the ability of the phage to infect different hosts. It also tells you that single amino acid changes, although the size of the perturbation is small, you can achieve large gain of function variants from this experiment. And it also highlights the power of deep sequencing to be able to resolve small functional differences between different mutations. Sorry, what's the time now? Okay. So I'll sort of briefly touch upon two additional vignettes here. One is we're looking at sort of metagenomic scanning of the receptor binding domain of T7. And phage evolution really happens in two steps, if you will. One is sort of single amino acid changes or small mutations, and we've already looked at that experiment, the data from that experiment in the last few slides. But phages also exchange large chunks of their genome by horizontal gene transfer, right? And so we wanted to ask, can we sort of simulate this experiment or this process through high throughput experiments? And so this is work done in collaboration with Karthik Ananthanaraman and in BACT and a talented student in his lab, Chris Kieft. And so I won't go into the details of the methods for how we do this, but the goal here was simply to ask, can we identify 6-mer and 10-mer sequence motifs from a large database of phage genomes? And so this phage database contains, thousands and thousands of phage genomes from all phage families. And what we wanted to ask was if we can insert these 6-mer, 10-mer sequences, and so these are defined phage sequences that we pick out, and insert them in different regions of the T7 genome, can we identify sequences that allows the T7 to infect hosts? And so the idea here was to sort of loosely simulate a horizontal gene transfer experiment. And so we experimentally synthesized about 11,000 sequence motifs. These are, again, 6-mers and 10-mers. And then we applied them on 5 different hosts. Again, the 3 hosts, the susceptible hosts, and 2 deletion mutants that we made. Now, we identified about 600 motifs that could infect at least one of the 5 hosts, which means that 95% of the sequence motifs essentially fall out or fail the selection, right, are not effective on any one host. But what's really neat is sort of this sequence specificity landscape that we can glean from this. So I'm going to walk you through this in a minute here. So the way this works is each column is a different host, right? And each row is a unique phage that carries a certain motif variant, right? And so they're clustered based on their functional profiles. And so what that means is if you look at this cluster 8, these are all the motifs which can infect all 5 hosts, right? And so now we can look at cluster 9. So these are phage variants that can only infect BW25113, right? And so you can go one step further and look at cluster 10. These are phage variants that can infect BW25113, 10G, and BL21. And I'll mention one more. Again, these are phage variants that can only infect BL21. So there's these really nice sort of specificity profiles of how these motifs can change the direction of the phage preference that kind of emerge from these experiments. And now what we can do is we can ask, well, where do these motifs fall on the receptor binding domain, right? And so here is, again, the sequence sort of 1 to 83. This is the PDB numbering. And so where the part is highlighted is where the motifs fall. So for instance, the cluster 8 sequences essentially fall in this region of the receptor binding domain. The cluster 9 sequences, which have this specificity, they fall down here. The cluster 10 motifs fall sort of flanking this region. And you can look at cluster 18, which is in the same region as cluster 8, but now you can go back and harvest the identity of sequences from these motifs, and they're actually quite different, right? Although they fall in the same region of the receptor binding domain. So this is still sort of data analysis that's ongoing, and so the goal here is to ask, are there sequences that are host specific and host agnostic? And are there motif sequences that are site-specific and site-agnostic, right? And so the idea here is to identify motifs that are both host-agnostic and site-agnostic. And the thinking is that these might be the sequences that are most exchanged across phages, and that's a bioinformatic question that we have to evaluate. So in the last couple of minutes, I want to sort of tell you our initial attempts at making therapeutic phages. So we focused on bacteria that's responsible for urinary tract infection. So UTI is a pretty major problem in the United States. 10 million office visits, 2 to 3 million emergency visits, a large number of hospitalizations, large healthcare costs. And the CDC report from a couple of years back said that community-acquired infection like UTI turns out to be harder to contain than hospital-acquired infection. And so we got some strains from Dr. Rod Welch here in MMI. And so what we did was we started combining. So before that, I want to say one quick thing, which is that I mentioned before that the efficacy of antibiotics is much higher than phages. And one way this manifests in terms of lower efficacy of phages is that there's rapid onset of resistance, which means that once you apply the phage, there is an initial drop in the bacterial load, but there is some resistant mutant that emerges, which kind of quickly takes over the population, and then the bacteria makes a full recovery. And so what we wanted to ask was, can we, now that we've done many selection experiments on different hosts, can we start combining beneficial mutations from multiple experiments, resynthesize a library, which is a combinatorial library of these mutations, and apply them on these pathogenic strains and ask if we have an effect? And so I'll just quickly mention one example here. So this is a UTI strain that we got from Rod. And when you have no phage, again, the bacteria kind of just grows fine and it stays the way it is. When you apply T7, there is a drop, and then after 8 hours, it kind of picks up, and then the pathogen, the bacteria, sort of falls off. And when we apply our mutant library, which has sort of combinations of mutations in the receptor binding domain, it's pretty close to a flat line. Now, there have been other instances where this doesn't work, so I'm just showing you the data that looks good, but there have been other UTI strains where these mutations don't work. And I'll mention one more example here, which is, so we want to sort of go for the worst offenders. And so if you look at the CDC website, there is the urgent threats, and there are the other groups. And one of the members of the urgent threat is this carbapenem-resistant enterobacteria, right? So some of these are the NDM1 strains, which are also called as superbugs. And so we got some of these from ATCC. And so the one that we worked on was resistant to 35 antibiotics based on the ATCC spec sheet, and was only susceptible to 2 antibiotics. And so now we again take these phage variant libraries and we apply that on this strain. And so again, no phage, there is sort of regular growth. When you apply wild type E. coli, there's the resistant strain that emerges. When we apply the T7 mutant, there is a resistant strain that emerges, but then it kind of falls off. And so again, this is not a clean kill, but it looks like even a naive experiment where we're combining mutations in one domain of the phage is giving sort of at least modestly promising results. And so the lessons that we learned from these experiments were that we think we now have a rapid pipeline for generating synthetic phages. And there have been a number of instances where these phage variants do outperform wild type, both in terms of the sensitivity and in terms of the efficacy. And again, based on some early results, we think that there's a path forward to test other multi-drug resistant strains and other pathogens. So I'll just close out by saying, we're pursuing a few interesting directions in the phage work in my lab. One is we now have a ton of sequence function data, and we really want to start making sense of that. And so we're using machine learning tools to ask if we can use the sequence function data to figure out the fundamental molecular principles in terms of amino acid mutation preferences on different hosts that govern specificity. We're working on different pathogens, and I mentioned a few here, but we're also working on pathogens that make thick capsules. And so these are pathogens that these capsules can evade the host immune system. So we're asking if we can engineer phages that can disrupt capsule-forming bacteria. We're super interested in using phages as tools to precisely edit the composition of microbiomes. And so the goal here is to ask if there is a specific bug that's offending, which is in the microbiome, which produces a certain metabolite that has some distal impact on some human tissue, can we use phages to edit out that bug? Now, what I've only mentioned so far is just mutations on the phage side. We're also working on high-throughput methods to interrogate mutations on the host side, to ask, where is the Achilles heel of the bacteria? So I want to go back to the question that I posed at the very beginning, which is, do we now know what's the molecular basis of protein function? I think it's fair to say that the answer is no. But I think what we can perhaps say a little more confidently is that we now have the methods in place to do this, right? And so we're making progress. And so if I were to go out on a limb, I'd say, maybe even the next decade. And so a good example here is how AlphaFold used a ton of information from PDB structures to crack the sequence to structure code. What we're hoping is once we have these large-scale sequence function data sets, new computational tools can start using these to hopefully crack the sequence to function code. And hopefully this will then lead to sort of exciting new mechanistic questions for others to pursue. With that, I'm going to stop. Again, this is just amazing work done by people in my lab, and I just have the good fortune to be able to present this. I want to thank all the funding agencies, UW-Madison, NIH DP2 Award, GLBRC, US Army, DITRA, NIAID, USDA, the Gates Foundation, and the Shaw Scientist Award. Thank you all.
So thanks a lot, Vatsan. I'm sure that Vatsan would take questions. And so since we're both recording and broadcasting, if you raise your hand, I'll bring the microphone over to you for questions. So questions for Vatsan? Go to Brian here first, since I saw his hand first.
Thank you. So I'm interested, your glycine to arginine mutation that's in the phage. You remove the receptor that would allegedly bind to. What does it go after next?
So, one hypothesis is there are secondary receptors that the phages can engage. So there's a preference for LPS, but there are maybe some outer membrane proteins like OMP that it can engage. So the thinking there is that maybe it's going in that direction. But again, one would have to do the knockout experiments to figure out what the other receptor is.
So just not known right now.
No.
Isn't.
Alan, you had a question?
So, Vatsan, when you do your screen across all of the amino acids in your 83 amino acid segment, it's a very non-evolutionary approach in the sense that you're always starting from wild type. And, Sean Carroll very nicely said in one of his books that evolution is like compound interest. You're always selecting on the next one and the next one. And so small changes in fitness translate into huge changes with so many cycles. So can you do something like that with your screen? Can you detect small improvements in fitness and then use that next mutant as the basis for selection?
Yeah, so we're doing that already. So we're again, we're using a little bit of machine learning here, but the goal is to ask, can we combine these beneficial mutations to make 3, 4, 5 mutation combinations that work really well? And the answer is yes. I haven't presented those data, but it actually does work. And so some of the pathogen kill curves that I showed were combinatorial mutants, not single mutants. I'll mention one more thing in this context is that it also turns out they're not just beneficial, there is very high epistasis. And what that means is you can have, say, 4 mutations that have sort of small fitness gains, right, 10%, 20%, and somehow the combination of 4 is like 1,000-fold higher than so this is case of extreme epistasis, and we've seen that too.
Other questions? I see one in the back.
So following up, is it on? Following up on your answer.
Yeah, it's just really weak. Hold it really close.
I can hear you.
Yeah, I'm not sure that the microphone is actually on.
It's not on, or at least
Okay.
it's weak.
Following up on your answer, so early on you mentioned exactly how broad the sequence space is. And I wonder whether you've started looking at using machine learning to use the existing sequence space to give you a sense of new starting points that might put you into an advantageous realm at the beginning of one of these processes so that you don't have to make so many combinatorial mutations.
Yeah, that's an interesting question, and the reason that's interesting is we initially tried to blast search to look for homologs to see if we can use that information. It turns out that there is like 50 hits which are 90% sequence identical, and then it just falls off the cliff. There's just no matches, which just means that the sequences diverge so far away that you can't pick up these patterns. But yes, for a different phage, for a different candidate, the approach that you're suggesting might work.
Jason?
Well, anyway, so Vatsan, I mean, it was a great talk, really interesting stuff. I like how the phage fitness landscapes vary by the host that you use. And is there anything in particular in the genotypes of those hosts that might indicate what's going on with the interaction between the phage genes and the host genes?
I mean, we have. So I mean, that's a question that we've thought about. But it's a tough one to answer because we have the genomic data, right, for all the hosts. But the question is, what do you look for, right? I mean, there are differences clearly between the hosts, right? So it's an n-body problem on this side and an n-body problem on the other side, n being the number of mutations and the number of genes on the host. So it's just to make that any causal connection is just, it's very, very hard to do.
Okay, can you hear me?
Yeah.
So 10 to the 31 phages?
Yep.
So do these fall into different categories? I'm wondering if they're different classes that you would use the same method to go after to expand your therapeutic potential. So would you go after a different class of phage for a different class of bacteria? And what is the scope? I don't know if that's bacteria space or phage space.
Yeah, I get the question. Yeah. So my imagination is that you clearly don't have to go for thousands of phages, but if you pick the largest pathogen groups, right? So let's say there are 6 or 8 or 10 of these, right? For each of them, you want to have some number of phages that you can rapidly evolve, right? And so the number is actually a fairly small number, right, in terms of the phages that one would have to modify to figure out these connections. It's just that you have to have the tools to be able to rapidly do that. So if again, I'll go out on a limb and say this, that we're looking at hundreds of phages. That's all we need, right? And so there's really nothing, if you ask, why is T7, why was that a good candidate? Well, it was really not a good candidate for any reason. It just happens to be the one that people have worked most with, right? And so there's a lot to be gained from working with known phages, in my opinion.
All right, one more from Brian here.
So what happened in this experiment where you fed one of your variants and the bacterial population was killed, then it came up, and then it went down again?
Yeah,
What's changing there?
Yeah, the answer is I don't know, but it's yeah, I don't know what's happening. So the one I can speculate what might be happening is, so the pool the wild type is clonal, right? The pool that we apply is hundreds of variants going in, and so one possibility is, there is a resistant bug that emerged, but some of the other guys in the mix were able to get to that, right? And so they were maybe dormant on the wild type pathogen, but once that evolved, the other guys could get to that. Again, but this is just hypothesis. We haven't demonstrated this. But that is the power of having a large cocktail of variants, because if one misses, the other one gets you.
So Vatsan, maybe I can sneak in one question here. I don't know if we have any others. We'll see. I'm curious, so where you're looking at the mutational landscape in different bacteria and seeing different extents of selection, if you will, different amounts of evolutionary pressure leading to greater or lesser amounts of selection for certain residues. I mean, some component of that has to be how many cycles of growth and pressure have been applied, right? And so the longer you go, the more something that's weakly deleterious drops out. So how do you think about that when you're comparing different infections, right? How do you decide that how do you standardize from one infection to the other to decide what the amount of selection really is?
Yeah, so so that's actually a tricky one to answer, only because, again, different strains are susceptible to different degrees. Now, in this case, so we have to maintain a balance between information and just complete wipeout selection that gives you one variant, right? So what we do is we ask, how long does it take for wild type to lyse this? And we'll give it a few multiples of that for the weaker ones to still have a chance. How many multiples that is almost a subjective question. But in this case, we applied 4 rounds of selection across the board. So they're apples to apples. But why 4 and why not 8? I mean, that's a hard one to answer, right?
Yeah, well, but a different lineage, depending on how dispersed it is, could be a different number of generations, if you will, in each of those passages, right?
And
something grows faster than another.
So the wild type is a useful reference all the time. So if it takes the wild type an hour to lyse, then we'd say we're going to give it 3 hours so that the weak ones can catch up. But there's nothing sacred about that multiple.
Other questions?
There's one hand in the back there.
Sorry, not seeing. Raise your hand again. Okay, sorry.
So coming back to Brian's question, again, I wonder whether there's a way to use essentially a synthetic recombination. You used recombination in your oracle to put in the proteins that you wanted, but can you use that against a larger set of, let's say, natural variants that you then can interrogate in different contexts?
A larger set of variants.
So like the natural variety that exists, because phages are constantly exchanging genetic information themselves. Can you impose a synthetic recombination strategy?
I mean, so one approach that might address this question is you can create a mutagenized set of T7 by radiation or whatnot. So now you have this sort of pseudo population of genetic variants, and you can measure the distribution, the extremities, right? And so you pick some multiple of that, which is perhaps a more realistic measure of how much deviation from wild type you want to have. So it's a pseudo population against an engineered mutant population. So that might be one way to do it. Right.
Any other questions? If not, let's thank Vatsan one last time for a really intriguing seminar.